Imagine a future where nobody dies waiting for a life-saving organ transplant. This future is now within reach thanks to lab-grown organs. In 2011, Sarah Murnahan received a double lung transplant after a long wait. Her story highlights the critical shortage of organ donors. Lab-grown organs offer hope for patients like Sarah. Grown using a patient's own cells, they eliminate rejection risks. Imagine a world where failing organs no longer rely on donor matches. Lab-grown organs could revolutionize healthcare and save millions.
Stem cells can develop into many different cell types in the body. They are like blank slates, transforming into specialized cells such as heart, liver, or lung cells. This ability makes them crucial for regenerative medicine, which focuses on repairing or replacing damaged cells, tissues, and organs. Different types of stem cells have unique properties and therapeutic potential. Induced pluripotent stem cells, or iPSCs, are created by reprogramming adult cells into an embryonic-like state. Shinya Yamanaka's Nobel Prize-winning discovery of iPSCs has revolutionized regenerative medicine.
The journey from a single stem cell to a fully functional organ is a complex and intricate process that requires a combination of cutting-edge technologies and a deep understanding of cell biology. One such technology at the forefront of organ regeneration is bioprinting. Imagine a three-dimensional printer that, instead of ink, uses living cells to create complex structures. This is the essence of bioprinting, a revolutionary technology that allows scientists to print tissues and organs layer by layer, creating three-dimensional structures that mimic the complexity of natural organs. Bioprinting relies on a bio-ink, a specialized ink made of living cells, and a biocompatible material that acts as a scaffold, providing structural support for the cells to grow and organize. The bio-ink is deposited onto a platform in a precise pattern dictated by a computer-aided design model of the desired organ. As the layers of bio-ink are printed, the cells begin to interact with each other and the scaffold, forming intricate networks and mimicking the natural organization of tissues. Another crucial aspect of organ regeneration is tissue engineering, which involves creating functional tissues and organs in the lab by combining cells, scaffolds, and growth factors. This interdisciplinary field draws from principles of biology, engineering, and materials science to create environments that mimic the natural environment of tissues and organs, allowing cells to grow, differentiate, and organize into functional units.
The field of lab-grown organs has already witnessed remarkable achievements, demonstrating the immense potential of this technology to transform healthcare. One of the earliest successes in organ regeneration was the creation of a lab-grown trachea in 2008. A team of scientists led by Dr. Paolo Macchiarini at the University of Barcelona successfully implanted a bioengineered trachea into a 30-year-old woman suffering from a rare airway disease. The trachea was created using a scaffold made from the patient's own donor trachea, which was then seeded with the patient's own stem cells. In another groundbreaking achievement, researchers at the Wake Forest Institute for Regenerative Medicine successfully implanted lab-grown bladders into young patients suffering from bladder dysfunction. The bladders were grown using the patient's own cells, eliminating the risk of rejection and restoring bladder function. These early successes, while significant, represent just the tip of the iceberg in the rapidly evolving field of organ regeneration. Researchers around the world are working tirelessly to create more complex organs, such as hearts, livers, and kidneys, bringing us closer to a future where organ failure is no longer a death sentence.
The process of lab-grown organs must adhere to strict ethical guidelines to ensure the safety and well-being of patients. Researchers must navigate complex ethical landscapes, balancing innovation with patient rights and societal norms. The road ahead involves continuous dialogue among scientists, ethicists, and policymakers to address these challenges and pave the way for responsible advancements in organ regeneration. As we stand on the brink of a medical revolution, the future of regenerative medicine holds endless possibilities. Imagine a world where organ failure is no longer a death sentence, where patients receive personalized organs tailored to their specific needs, and where the scarcity of organ donors is a challenge of the past. But the journey is far from over. The path to widespread adoption of lab-grown organs is fraught with scientific, ethical, and regulatory hurdles. Yet with each breakthrough, we move one step closer to a future where the boundaries of human health and longevity are redefined. As we envision this future, we must remember that it is shaped not only by the advancements in technology, but also by our collective commitment to ethical integrity and compassion. The dream of eradicating organ shortages is within reach, but it requires the collaboration and dedication of scientists, policymakers, and society as a whole. If you liked the video, subscribe to the channel and let us know in the comments below. What do you think the future holds for regenerative medicine? Together we can make this future a reality.